and we'll talk about some population genetics. So when I took genetics, this was my least favorite topic, and since I've begun teaching it, it's become one of the ones that I actually started to really enjoy more. It's starting to see that its significance, because it's a pretty significant topic in genetics. Yeah. Okay. So, and yeah, somebody asked, yes, the lowest test score is getting dropped. So I had said that at one point. I don't know if I made an actual announcement about it, but the lowest test score will figure out your grade based on the average of your 3 best test scores plus the other assignments, and they will still count towards 80%. Test will be 80% of the grade. So the percentage-wise doesn't change at all. Just we will drop one test. All right, so onward. Excuse me. All right, so the thing about population genetics, it's different, is we look at how genes behave in populations. And we want to understand that there are certain alleles in a population, what governs like how they're distributed throughout the population. How does it determine phenotypes? What factors might change this allele distribution? And a little bit about how detrimental alleles can persist or increase in populations. So things some terms just to start off. A population basically is a group of individuals. You can define populations very broadly. You could say population is the population of the United States or the population of Europe. You could say the population is the town you live in. You can come up with different ways to define a population, how big it is. The gene pool is just the complete set of genetic information in all the individuals within a population. Phenotypic frequency is a proportion of individuals that have a phenotype, so something you can observe, and the genotypic frequency is what's the proportion of individuals in the population that have a genotype that give that phenotype. Because we know that certain phenotypes can have more than one genotype. Classic case of dominant and recessive alleles, you can be homozygous for the dominant allele or heterozygous for dominant and recessive allele, but still have the dominant phenotype. So these genotype and phenotypic frequencies, of course, are different. And then we'll start talking about allele frequency, and that's proportion of any specific allele in a population. So how much does an allele exist in a population? So there are a couple fundamental calculations that we have to do. One is the allele frequency, and that's a good place to start. The allele frequency is the number of copies of an allele in a population divided by the total number of alleles for that gene in a population. So, for example, the number of dominant alleles, let's just say gene A, dominant A alleles in a population. But what's the total number, of those alleles versus the total, possible alleles that there are in a population? And then there's the genotypic frequency. Now we're going to look at individuals, the number of individuals in a particular genotype in a population divided by the total number of individuals in a population. So let's go through some of these. So we're going to consider a hypothetical population of 100 frogs. Now, these frogs have different degrees of greenness, and they can be determined by by their genotype. So you can look at their phenotype, and you will immediately know their genotype. So we have 64 dark green frogs, 32 medium green, and 4 light, and those are designated. The dark green frogs are, we'll say, capital G. The heterozygous frogs are the medium coloration, and the light green frogs are homozygous for the little g allele. Okay, so this is all kind of stuff you've seen before. If we start looking at the frequency of the little g allele, what's the frequency? So the frequency is going to be the number of 2 times the number of heterozygotes. So this component here. So, 2 times the number of homozygotes for that allele, because each one of these individuals has 2 alleles. So we're counting the alleles now, not the individuals. So in counting the alleles, each individual has 2 alleles. In this case, these are the homozygous little g alleles, plus all the individuals with that are heterozygous, because they have 1 of these little g alleles. And so we can say this will give you the total number of little g alleles in the population, right? 2 from these guys, each one of these, and 1 from each of the medium green frogs. To find the total number of alleles, it's simply the total number of individuals, and each one has 2 chromosomes. They can have 2 alleles. So it'll be 2 times the total individuals. So we go through and we have four of these that are homozygous for little g, 32 of the heterozygous individuals, 2 times, we already said there's the population is 100 frogs. So we get all these numbers, we plug them in, and we get a frequency of 0.2 or 20%. Does everyone sort of figure out that sort of the math is obviously very simple, but knowing how to define which phenotypes and genotypes count for which, how many alleles is important. Okay, so we can go through and do the same thing for the big G allele, or it is the very same fashion. Or it's much easier if we just know that for any given trait, the allele frequencies is always going to be less than or equal to 1, because that represents 100%. Can't have more than 100% of your alleles being anything. And so for alleles with, for genes with more than 1 allele, the frequencies of all the alleles should add up to 1. So an easier way to do this is just to say, well, we know that this is the frequency of the little g allele, 0.2. We can just subtract that from 1, and we get 0.8 or 80% of the alleles are the big G allele. So again, very simple. So why am I going through this? This is very simple. This is when we're trying to figure out sort of the allele frequencies when we don't have such a nice distinction between dark green frogs and medium green frogs, which are homozygous for the big G allele or heterozygous. It becomes just slightly more complicated. Okay, so P is usually given, the designation is given for the frequency of the dominant allele, and Q is the frequency of the recessive allele, and P plus Q must equal 1. So that's everything that we've talked about up until now. And if you go to a third allele, it's sort of in the ABO blood types, that would be R. And then P plus Q plus R should equal 1. All right, so you can, and then you can continue depending on how many alleles are there. You can keep giving them designations. But we're going to just focus on, typical 2 allele situations. And so we're going to continue using the frog example for a few slides, and the frequencies that we've already calculated. So we're going to just build and continue with that. We're going to talk soon about the Hardy-Weinberg equilibrium. Okay. So just again, this is again going to be I think fairly straightforward. Some of you might even be like, insulted. Why are you telling me this? This is a very easy example to figure this out. So if we want to figure out the frequency of the genotype here, it's the number of individuals in a population divided by the total number of individuals in the population with this genotype, which is divided by the total number of individuals in a population. And again, this is easy because our phenotype, we can observe a phenotype and know exactly what the genotype is, which also tells how many alleles are in that individual of a particular type. So this is a very simplistic example. Right, so simply for the dark green frogs, you would take 64 over 100. And for the heterozygous and the homozygous for the light green allele, you simply take the total number of individuals divided by the total number of individuals, with that genotype divided by the total number of individuals in the population. Again, very straightforward. All right, so now we can figure out how to tell how many alleles are in a population, what's their frequency, the different phenotypes and genotypes that are in a population. What sort of rules can we sort of come up with and govern this? Now, this has been worked out quite some time ago by Hardy and Weinberg, and they had a mathematical way to describe the relationship between the proportions of alleles in a gene pool and the frequency of the different genotypes. And in order for a population to be in what's called Hardy-Weinberg equilibrium, you have to assume that the population is ideal. Now, what does ideal mean? No population is going to be ideal, but there are certain assumptions that you can make, so to define this ideal population, and we can use their equation to sort of tell us what to expect if it was an ideal population, and then if it doesn't meet that, you can start looking and see, well, why doesn't it meet that? Okay, so first assumption is that the population is infinitely large. Now we know that not to be true, but very large populations do exist. And so this is something we'll talk about later about genetic drift, and having a very large population sort of reduces the effect of this phenomenon of genetic drift. Another component is that there's no migration into or out of the population. So the individuals in the population are pretty stable, and they don't move around. There's no selection. All the genotypes have equal viability and fertility, and that's not always the case, but there are lots of examples of that. So nothing is getting selected against or for. The frequencies of an allele are the same in males and females. Well, they might, you might have a X-linked trait. The alleles that are governed by that are still the same both in males and females. The frequency of those alleles. Where you negate sort of mutations, so assume mutations not occurring, so we're not getting new alleles coming from mutation, and that random mating occurs, that there's no preferential mating. And so that sort of figure, I'll use this sort of diagram later on, sort of talk about these different things. So we're going to talk about what's ideal, and then we'll talk about some things that make it less than ideal. And so when you make all those assumptions, you can make a couple predictions. The frequency of the alleles in a gene pool does not change over time. After one generation of random mating, the genotypic frequencies for two alleles can be calculated by this formula. P squared plus 2pq plus q squared equals 1. And again, P is going to be the frequency of the dominant allele, and Q is going to be the frequency of the recessive allele. These are going to be the homozygous, dominant heterozygous individuals and the homozygous recessive. And so how do we come up with that? So if we have a heterozygous mating, so this is a typical sort of F1 mating between any individuals. And so they have one dominant allele and one recessive allele. The frequency of the dominant allele is P, and the frequency of the recessive allele is Q. Now this is this is the mother and the father. They have the same. They're heterozygous. The frequencies are the same. So we can look now. We're used to looking at this. So with this individual here, we know this is how you get a homozygous dominant individual. When you have gametes formed and you get fertilization from a gamete that has this allele from the mother, and a gamete that has the same allele from the father. So this is all stuff that you've seen in the beginning of the class. But now we can talk in terms of the frequencies. So the frequencies are going to be P coming, or this allele is going to be P, but it's also P coming from the father. And so since these are, whether or not these alleles will come together and this fertilization is sort of random, so it will be the product of these two. It would be P times P or P squared, right? So P squared sort of defines the homozygous dominant individuals. And then the same is true if we look down to this quadrant over here for the same reasoning for the recessive alleles. So we get Q squared. And then for the heterozygous, we have two ways that they can occur. We have either getting the dominant allele from the mother and the recessive allele from the father. So that's going to be P times Q. Or the recessive allele from the mother and the dominant allele from the father, and that's going to be Q times P. But that comes out to be the same, so you have 2 PQ. So this is how this equation comes about. These are the homozygous dominant individuals, as I mentioned. These are the heterozygous individuals, and these are the homozygous recessive individuals. And all the frequency of these should equal 100% of the population, or 1. All right, so we've looked at this hypothetical population, and we've calculated the genotypic frequencies by calculating the allelic frequencies. And so we know that the calculated allelic frequencies are 0.8 and 2, and excuse me, 0.2. So if this population is in Hardy-Weinberg equilibrium, then using the allele frequencies, we should be able to calculate the frequencies of each of these different individuals. And so here, if we go through and we do the math, we get the numbers which are the same as what's here. And again, this is a hypothetical population, so this is supposed to happen. But what's key here is that, because this does not always happen, is that if the Hardy-Weinberg genotype frequencies that you get from the genotype frequencies that you get from the Hardy-Weinberg equation, for the components of the Hardy-Weinberg equation, equal the calculated genotypic frequencies, then the population is in Hardy-Weinberg equilibrium. So these numbers match within statistical error. Okay, questions on that before I move on? And I should note that this is done fairly well in the textbook. So you should definitely give this section a read. Okay, so I'm going to talk about sort of a case study having to do with HIV, or human immunodeficiency virus. So first, a couple things about HIV, which many of us have heard. It's a member of a retrovirus family. So it has an RNA genome. It causes acquired immunodeficiency syndrome, or AIDS. And for humans, it's a condition where the immune system begins to fail, leading to life-threatening opportunistic infections. A lot of people who die from this die from bacterial infections, and so it's the immune system is being compromised here. And so the four major routes of getting this are unsafe sex, contaminated needles, breast milk, and perinatal transmission. All right, so what do we need to know for this sort of population genetic segment? So HIV infects the primary vital cells for the human immune system, so helper T cells, specifically the CD4 plus T cells. It can also infect macrophages and dendritic cells. Now, we didn't talk about the immune system, and I don't think we're going to have time to this semester. But think of it. But keep in mind, what to focus on here is that these are cells in the immune system. It's these helper T cells that we're going to focus on. And there are certain receptors. There's CD4, and CD4 is a glycoprotein that's expressed. That's on the surface of the T helper cells and other cells, and CCR5, which is a CC chemokine receptor type 5, and it's an integral membrane protein that's on the surface of the cell. So it was noticed some time ago that there are a small number of individuals who practice high-risk behavior, so it's having unprotected sex with HIV-positive partners, who never get infected. So what is going on here? And so back in the mid-90s, they discovered that in 2 of these individuals, they were homozygous for a mutant allele in the gene that encodes this receptor, this CCR5 receptor. So this mutation was a 32 base pair deletion, and so this allele is called the delta 32. Delta oftentimes in genetics is used to indicate a deletion. So this is delta 32 is a deletion of 32 base pairs. Now in the figures and tables, the wild type allele is just called 1. So if we have an individual here that is homozygous for the wild type allele, it'll be 1, 1. They are susceptible to HIV. Heterozygous individuals are susceptible, but AIDS does not progress as quickly. Those individuals who are homozygous for the delta 32 allele are resistant to most sexually transmitted strains of HIV. Right, so here's the mutation. Here is the sequence of the CCR5 gene and the translation of the gene to the amino acid sequence is shown here. And this is where there's a deletion of these 32 base pairs shown from here to here in red. This deletion, since it's not a multiple of 3, creates not only this deletion, but a frame shift thereafter. So normally we'd have this threonine here, but now this becomes the first nucleotide, and the codons frame gets shifted. So now we have an isoleucine, where normally we would probably have a threonine. But again, this isoleucine is now juxtaposed next to this tyrosine because this whole part is deleted. The other thing that this frame shift does, which happens a lot in frame shifts, is it creates or exposes a stop codon, which was normally out of frame with the protein coding sequence, and now all of a sudden gets put into frame, and you get a premature stop of translation. And so normally in the wild type protein, this amino acid sequence would continue for another 122 amino acids. So this stop codon, this TGA right here, gets created, and it sort of makes a deficient CCR5 protein. Now, this can be observed through molecular techniques. We can do PCR, which we talked about, and restriction digests to identify the alleles that an individual has. So what we can see here is that if an individual is homozygous for the wild type allele, they're going to get a band of a certain size that represents the total gene or region of the gene that includes these 32 base pairs. And you get another fragment that exists in all of them. This comes from the restriction digest. That we're not going to consider this, but this is common regardless of the allele that's there. In individuals that are homozygous for the delta 32 allele, they have a smaller piece by, you guessed it, 32 nucleotides. And so you can distinguish the different alleles. And in an individual that is heterozygous, you can see both of them. So you can determine the genotype through molecular techniques. And so what's going on with this receptor is normally you have CCR5 and CD4 receptors, which allow HIV infection. When you have a mutation in this CCR5 receptor, it prevents HIV from binding, and the virus can't infect the cell. Okay. And so that's sort of what's going on in sort of molecular details of what's happening here. So now let's get back to what's going on in this population. Okay, so they surveyed 1,000 individuals from France, and they looked at the genotype frequencies. Now remember, they can determine this genotype because they can do this PCR and restriction digests to determine what alleles are there. And so they 795 of the wild homozygous for the wild type, 190 heterozygous individuals, and then 15 with this delta 30 that are homozygous for the delta 32 allele. And so I don't particularly like these designations, so I'm just going to switch to calling them, either capital A for the wild type allele or lowercase a for the delta 32 allele. So we can write something like this. We want to figure out the allelic frequencies that are occurring in this population. So how many of the wild type alleles are there? Again, they are defined by the number of individuals that are homozygous with the dominant allele times 2, because each individual has 2 of those alleles, plus all the heterozygous individuals because they have 1 dominant allele. This one allele here, divided by the total. So when we substitute in all these numbers coming from here, we get the frequency of 0.89 or 89%. We can go through and calculate this. We can either subtract or we can go through the calculation. I went through the calculation here. Again, counting up all the homozygous recessive individuals times 2 to get the number of alleles, plus again, the heterozygous individuals come into play here, divided by the 2 times the total number of individuals, and we get 0.11 frequency or 11%. Another question is, so we know the frequency of the alleles, of each allele, and then we can ask, is this population in Hardy-Weinberg equilibrium? So we determine the frequencies of the genotypes, we calculate the allelic frequencies and predict the next generation using the Hardy-Weinberg equation. All right, so this observed, so I should back up for a second. It's clear. These are the frequencies that are observed. This is what's happening in real life. So these come from the previous page, and the calculated ones are the ones that are calculated using the components in the Hardy-Weinberg equation. And so we get these. We get this number of 0.792, which simply comes from the allelic frequencies that we've determined, and it's as for p, as p squared. We get this number. We go through and do that for the rest of them, and we get these frequencies that come about from the Hardy-Weinberg equation from those that were observed, and we compare them. And so, without going through the statistics for this, these were shown to be within error. So these tell us that this population is in Hardy-Weinberg equilibrium. Now, an interesting thing about this is that if you take a subpopulation, so again, you can always redefine what population you're looking at. So the subpopulation of people with AIDS but are not really progressing, the heterozygote frequency is very high. 40% of those are heterozygous. And this is another sort of support that this delta 32 allele is giving protection from HIV infection. All right, so that's a way of using sort of population genetics with sort of a real case scenario. All right, so I mentioned, so before I move on, any questions on this topic? I found this fascinating when I first learned about this, and this is so. I mentioned how when I started out, I was never really interested in population genetics. When I took the class, I didn't learn about stuff like this. And so when I started learning about this, I found this fascinating and sort of started to get, increase my interest in the topic over the years. Okay, so what if you want to calculate these frequencies from phenotypes? And the phenotypes are not as simple as we have dark, depending on what the genotype is, we can see a dark green frog, a medium green frog, or a light green frog. We don't always have that option, right? So we have, a dominant phenotype can typically either be heterozygous for the dominant allele or homozygous individual. So that's going to be the frequency of people with or individuals with the dominant phenotype is going to be p squared plus 2pq. is composed of both allele frequencies, this is not a good place to start to try to figure out what allele frequencies are. However, if we look at those that have the recessive phenotype, there's only one component, it's Q squared. And this is something that we can really start to use to start solving sort of like these problems of what are the allele frequencies. So whenever possible, start with the recessive phenotype. Because this can be determined by individuals demonstrating the phenotype. We know that based on the homozygous recessive genotype, so we know what the genotype is for these individuals that have the recessive phenotype, whereas those for the dominant phenotype, we don't know what their genotype is. Start out assuming Hardy-Weinberg equilibrium, and then from this we can determine the allele and genotype frequencies of this population. All right, so here is an example. So PTC tasting. So this is, you may have done this as a kid, where you taste this paper and either it tastes bitter or it doesn't. You have a question?
Yes, Dr. Matagora.
Yeah. Yes.
This is Lee. On the previous slide, why are we worried about the specific genotype of like heterozygous dominant versus homozygous dominant if all we're focusing on for this slide is the phenotype?
So we're going to focus on, we're given the phenotype, because phenotype is what we can see, right? We've talked about situations where either the phenotype equals the genotype just visually with the frogs. We talked about the CCR5, sort of phenotype. We can see the genotype by using a molecular technique, but what if you can't? What if it's not that simple? You still want to find out from a population standpoint, what are the allele frequencies? But all you can see are the phenotypes. So you can still get to the allele frequencies. It's very difficult to go through the dominant phenotype because there are two ways that an individual can have the dominant phenotype. And in doing so, we end up having the frequency of the dominant allele and the frequency of the recessive allele in the equation. If we want to determine the allele frequencies, we'd have to solve for P or Q. Whereas if we just look at the recessive individuals, there's the only frequency that there is the frequency of the recessive allele. With the phenotype, we know what the genotype for that phenotype is, and we know what the frequency, how to define the frequency of those alleles. It's going to be Q squared, because all we have are recessive alleles. So this is on a pathway of getting to the allelic frequencies. So you're calculating these allelic frequencies from phenotypes. Does that make more sense?
Yes, that actually does. And I realized that this slide is actually focusing on calculating the gene frequencies from the phenotype.
Yeah.
I had thought that you were calculating the phenotypic frequency.
Oh, no. We're going to eventually get to the genotype, get to allelic frequencies, and then get to the genotypic frequencies. Yeah.
Okay.
It's a little bit of a, but it's actually a little bit of a confusing header there. I never really thought about that. Okay.
Thank you.
Hopefully this example will sort of help, with sort of that question. Right. So here we have our phenotype is like whether or not an individual can taste like this bitter taste from this PTC test. I've forgotten what PTC stands for. I don't remember. I think I did this when I was a kid, and I don't remember what I tasted, but I imagine that I didn't taste it because people say when you do, you don't forget. So this is a dominant allele governs this. So whether you can taste it, if you can taste it, you have a dominant phenotype. So if you have 70% of the population are tasters, they can taste this, right? What does that tell you? How do we sort of figure out, our allelic frequencies from that, if we want to know this, and then to determine what are these allele frequencies? So again, we'll make the assumption of Hardy-Weinberg equilibrium. I'm using capital T for the dominant allele, lowercase t for the recessive allele. So this is what I was saying. If we look at the frequency of the dominant phenotype, which can be defined by two different genotypes, we get this equation. So this is equal to 0.7. So going through and solving this is not going to be simple. However, we know that the frequency of q squared, that we can look at the frequency of q squared, which is going to be 1. So we rearrange the Hardy-Weinberg equilibrium equation. It's going to be 1 minus all of this, which has been defined at 0.7 from 70% of the population. So we know the frequency of the those with a recessive phenotype is 0.3 or 30%. That becomes pretty obvious when you sort of look at this. This is a very convoluted way to say, like, oh, why don't I just subtract this 70% from 100% to get how many recessive individuals there are? But remember, so we're going to come up 0.3. So that works, and this is why. But remember that this is represents q squared, so it's going to be the square root to get the allelic frequency. To get the frequency of q, you just need to take the square root of these two. And I don't have a convenient little square root character to show this. So it's this is 0.3 to the half or square root. And so the frequency of the recessive allele is 0.548. Now we can take this, plug this back into, since we know p plus q equals 1, and now we've just determined what q is, we can figure out that the frequency of the p allele is 0.452. So this is a way to get down to these allelic frequencies when you're just given the frequency of a phenotype, of one phenotype. And in this case, it's the phenotype that's it's the dominant phenotype, which is more difficult to deal with. You just need to subtract that, in this case, from 100%, and you get 30% or 0.3 for frequency, and then go through and substitute that in here where it's appropriate, and calculate the frequency of the recessive allele, and from that you can calculate the frequency of the dominant allele. Okay, does that make sense to people? Any questions on that? Looking good so far. All right, so for X-linked genes, frequency of the X-linked allele in gene pool will equal the frequency of the males expressing that X-linked trait. Remember, the males, if it's a gene on the X chromosome, they only have one copy of the X chromosome, and so whatever allele they have here will be expressed. So for those with the dominant phenotype, they're going to have the capital A allele. Those with the recessive will have the lowercase allele, and this is going to directly the frequency that we see for the individuals with for the males with a particular phenotype is going to equal their genotype, because we know what that is, which will equal the allele frequency. It is more complicated when looking at females, because they have two copies of each allele. So we'd have to go through the same business that we went in the last problem. And so this is where this assumption, this Hardy-Weinberg assumption comes into play, that the allelic frequencies are the same in males and females. So as long as that is true, if we can calculate the frequency in the males, it's then going to be the same frequency in the females, because we are making this assumption. Okay, so classic example of this is for an X-linked gene is color blindness. And in American males, 8% of American males are red-green colorblind. And so we'll designate the uppercase C for normal vision and lowercase c for color blindness. And so we know that since 8% of the males have this, if we look at only the males, that means Q is going to equal 0.08. Right? Because in males, this is the phenotype frequency is the same as the allelic frequency. So then we can calculate what the frequency of the uppercase C allele is, which is 0.992. And so we can look at what so we stop right here. We now can easily define what the allelic frequencies are for this X-linked gene. That's one of the easier things that we can do with this. But now if we want to go see, ask the question, what fraction of the females in a population would expect it to have this trait? It would be Q squared, or 0.64%. So it's much more rare to have females with this type of color blindness. All right, so we mentioned the three consequences of Hardy-Weinberg law is that the dominant traits do not increase from one generation to the next simply because they're dominant. Genetic variation can be maintained in a population because nothing really changes. Knowing the frequency of just one genotype will allow us to calculate frequencies of all other genotypes. And this is important for calculating the heterozygous carriers of recessive genetic disorders. So I have a question on the previous slide of how much more rare is in females. This is the males have 8%. So I think the person who's asking this is not 64%. This is 0.64%. So it's much lower than the 8%. This is not 64%. It's 0.64. Okay. Okay. So all right. So this is sort of Hardy-Weinberg. Things are in equilibrium and things don't change. But how do they change? It has to be some sort of violation of all these assumptions that we are making. So in real life, violations happen all the time. And so there are different ways to violate this: genetic drift, mutation, selection, non-random mating, and migration. So we've mentioned these previously. And these are going to have gain or loss or just changing the frequency of particular genotypes. And what's important about this is how a population can evolve, how it can change from having certain characteristics in one population and then evolve to gain new characteristics. And so the first thing we'll talk about is selection. And the most extensive one that we talk about, because there's lots of different types of selection. So everyone's heard the term natural selection, and let's sort of talk about what this means. So first we're going to talk about fitness. So Darwinian fitness is a measure of reproductive superiority. How well can having certain traits will promote a individual to be able to mate and pass its genes on to the next generation. And so it's the relative likelihood that a genotype will survive and contribute to the gene pool of the next generation. And we should not confuse this with any form of physical fitness. That's not what what we're talking about here. They say survival of the fittest is not necessarily those that are the most physically fit. It's the individuals that have the traits that will allow them to pass their genes on to the next generation. And so differences in reproductive fitness can be due to fittest genotype is more likely just to survive. This genotype will be more likely to mate, or more likely, or also it can be that they are more fertile. So there are different reasons that an individual can have reproductive fitness, or different levels of reproductive fitness. And the thing here that I sort of emphasize that natural selection, this is the major force that shifts allele frequencies in large populations. So this is really what's governing the shift of frequencies. And so we're going to talk about some different kinds of natural selection. And we're talking about directional selection, balancing selection, disruptive or diversifying selection, and stabilizing selection. Okay, so let's start with directional selection. In directional selection, genotype that gives a particular phenotype is selected, resulting in a change in the average of this in the population of this frequency. So here we have butterflies with light wings, and we have mostly butterflies with light wings, and then some event comes along that, and so we can see the graph here. Most of the individuals have light wings, and then over many generations, there's a selection in a direction to increase the number of butterflies with dark wings. So this is a directional selection. In this case that we're talking about, we're starting with population has light wings and moving to a population with dark wings. And this is going to be due to some environmental fact. Example that's easy to, that we've given some, this goes all the way back to the very first lecture. This dark coloration is favored when to help butterflies sort of avoid predation in forested areas. So if the population moves more towards something drives them towards the forest, those with the darker wings are going to survive better. This can also go in the reverse direction. If they start out with a population with darker wings, you can imagine deforestation, right? Where most of the population is in a forest and the forest gets removed, then the ones with the darker wings are not going to survive as well. They're going to be, be eaten by birds, more often or something like that. And the population will then could shift going back in the other direction, and where those butterflies that have lighter wings will survive better in the sort of unforested regions. So there's a selection in a direction, and this is the scenario shown here goes from light to dark, but it can easily go from dark to light. So we have balancing selection. So there are two alleles that reach an equilibrium where opposing selective forces balance everything out. The population isn't just moving towards allele fixation or elimination. So I should have defined these terms in here, and I think it came up this early. So allele fixation means that if you start out with a population with a certain number of two alleles for a particular trait, an allele that is fixed means that it is represented in 100% of the individuals. So that's the only allele that's there. And allele elimination, you can imagine what that is that now all of a sudden there are none of these alleles in the population. And we talk about allele frequencies. Fixation has a frequency of 1, or 100%. And when an allele is lost or eliminated, it has a frequency of 0. And this is a way so in sort of the directional example that I showed, you start moving, you start reducing one of the alleles, start moving from from one population here with light wings. You're losing the light wing alleles and moving towards more dark wing alleles. If this was to go to completion, could have loss of the light wing allele, and then we couldn't reverse. Usually that doesn't really happen. I mean, that's not a fair statement. In the example here, usually you'll have some of the alleles kept in the population, but there are ways that you can lose alleles. In balancing selection, we're not moving in that sort of direction. That not moving to predominantly have one allele or the other. This maintains more than one allele in the population. we saw this in an example of heterozygote advantage, where there's a better chance of survival of the heterozygote individual than either homozygous individual. And the example that we talked about was sickle cell anemia, where we have this individuals who have this the S allele, homozygous for this, will have sickle cell anemia. Those that have the A allele are normal, but they're still susceptible to malaria. Whereas the heterozygous individuals are prevented from having the sickle cell anemia, but they're also resistant to malaria. So this is sort of a balancing selection, keeping both alleles are being kept in the population. Another way that this happens is what's called negative frequency dependent selection. And this is common individuals lose fitness simply by the fact of being common, so being more abundant. Rare individuals gain fitness because they are rare. And so selection always favors the less numerous genotype. And this will drive the selection towards a balance. So what does this mean? The example here is this example for this involving a rewardless flower. So this is a flower that has no nectar. So pollinators will fly around and go to different flowers and try to get nectar out of them. But they're going to learn that a flower with a certain color, the most common of flowers. So none of the flowers have nectar. But they're going to run to, say, for example, say yellow is most common. They're going to run to the yellow flowers. They're going to learn that yellow flowers have no nectar. So they're going to start avoiding those. And when they see a different colored flower that's more rare, they're going to be hoping to get nectar from those flowers. And so now they're going to start pollinating those flowers. They're going to avoid pollinating, in the case that I mentioned, the yellow flowers. They're more likely to pollinate the red flowers. So what's going to happen is the red flowers are going to increase in number over time, and then they're going to become common. And then the pollinators are going to avoid those and go looking for the next one. So it just starts all over. And it's cyclical, is that they start, there's a lot of one type of flower, and that's going to decrease because now they're saying, like, wait a minute, they're learning these have no nectar in them. What about these? They start pollinating these, and they start to increase, and they get to a point where, wait a minute, these have no nectar. Let's go take a look at the other ones. And they start, these come down, and then the other ones go back up, and so they just oscillate back and forth. And so this helps to keep both alleles governing color of this flower in the population, because when one becomes too common, it loses advantage. The pollinators are not going to look to it for nectar, so they're not going to pollinate that one as frequently. Okay. Okay, so just another form of selection is disruptive selection. This favors the survival of two or more different genotypes with different phenotypes. It's also known as diversifying selection. This is caused by fitness failures for a given genotype that vary in different environments. So this is going to be environmentally based. This will result in a population with an increasingly bimodal distribution for the trait. So here we have our starting population. There's a certain number of individuals with a phenotype, and on the X we have just a spectrum of different phenotypes that can occur due to this trait. Depending on the environment, if the environment changes, there will still, if the environment, if part of the environment remains as it originally was, there'll be individuals that have a genotype that allows them to survive very well in that environment. The new environment will have individuals that, very few on this graph exist, will now start to have an advantage and will reproduce more effectively in this new environment, and we'll start to see more of those individuals occurring. This is a case where this goes to more extreme level. This is sort of indicative of what happens in speciation. You start with an original population, and now you start to get divergence of these of two different populations based on two different environmental regions, and eventually these could become new species. Okay, and then the last one I'm talking about is stabilizing selection. This is where having extreme phenotype is selected against, and only those things that are those that have phenotypes that are sort of in the middle range will survive. They have the highest fitness values. So one's birds' clutch size, or the number of eggs in a nest, so it's called clutch. Too many eggs will drain the resources that the adult birds need to care for their young. Too few eggs means that they may not contribute many individuals to the next generation. Some might, if you have few eggs, they can easily get eaten by a predator, or just even if they all survive, there's not enough to really make as huge of a or as big of a contribution to the next generation. So if a population starts out with the number of eggs, sort of a range of number of eggs, over time, this is going to get a more tighter curve, that the population is going to only have a specific number of eggs, like in a very small range. The extremes don't occur very frequently. And the result of this is less diversity. This is more uniform, whereas the disruptive selection is actually, we've just seen, increases the diversity. Now, another example of this is human birth weight. So if we have birth weight going along the X, and we have mortality. So if we look at birth weight first, we see that most babies when they're born are around 7 pounds, between 6 and 9 pounds. But there are very few that are born at the extremes, and that's because the smaller birth weight, there's a high degree of infant mortality. So these individuals just don't survive, and that drops precipitously till we get down to an average of around 7 pounds, but then starts to increase again as the birth weight increases. So this is an example of stabilizing selection. So the extremes are, phenotypes are get eliminated, and we have really just this sort of moderate or middle number. In this case, birth weight that survives. Okay, so that's the most powerful force in driving the evolution of a population. Any questions on selection?
Dr. Bonacora?
Yes.
For the stabilizing selection,
Mm-hmm.
both the clutch size and the amount of eggs being hatched or being laid, are selected at the same time, or is there a correlation between the two?
Well, they're only going to lay, so clutch size, they're only going to lay a certain number of eggs. All right, so they're going to the birds are not going to here that they're at this point in the population, those that lay like too many eggs, they will not they will have less chance of surviving and reproducing in the next generation. Those eggs may not make it to the next generation. So it's I guess you say they're correlated, so they're related to each other. Does that make sense?
Yes, thank you.
All right. So now next feature to talk about, and this one is mutation. So we've talked about mutation, and mutation provides the raw material for evolution, but it actually plays a very small, insignificant role in changing allele frequencies. Here, if we have an example where we have the frequency of as we call it, the big allele is at 100%. Now, in order for that to change, it has to change by mutation. And if it's mutation alone, the frequency of mutations is very low, and this gets determined experimentally. But what happens is it will take 70,000 generations just to reduce this allele frequency by mutation alone from completely fixed at 100% to 50%. Now, that's if the mutation is not selected upon. It's just a mutation that has no effect on the on any selection. If the mutation arises that gives a selective advantage, one of the things that we mentioned before, gives some sort of selective advantage to that population, we'll start seeing that those individuals that will get passed on more frequently, and this can get this sort of this graph can become more steep, but that's if there's a selection for it. So the frequency could increase more rapidly. But if we're just talking about each time whether or not we're going to get a mutation to increase the frequency, well, the mutation has to be the same, give the same phenotype, and it's not occurring that quickly. It doesn't contribute that much. Something that can contribute to changing allele frequencies is migration. So the idea is just movement of some portion of a population from one place to another. An example I have here, there are two populations, one on the mainland and one on an island. And so some portion of the mainland population moves to the island. This is going to change the allele frequency. And the degree that it can change that depends on a couple of things. It depends on the rate of migration. So if there's a lot of individuals from the mainland move to the island, well, now they're contributing a whole bunch of alleles to this new population, or to the island population, and that can really have a huge effect on the allelic frequency. If only a few individuals move, it may not have that big of a. The other thing is that makes a big effect is the allelic difference in the allelic frequencies from those on the mainland to those on the island. If these individuals have a very different frequency of each of the alleles for a particular trait on the mainland compared to the island, there'll be a more pronounced effect. So it will have a greater effect on what the final allelic frequency is on the island. Think of it the other way: if the allelic frequencies are exactly the same on the mainland and on the island, it doesn't matter how many move from the mainland. It's not going to alter the allelic frequencies. There has to be some sort of difference, and the greater that difference between the allelic frequencies, the greater the effect migration will have. And now we come to my favorite one, which is genetic drift. Genetic drift is also called random genetic drift, and the randomness is something that I've always just appreciated. And genetic drift has an effect on small populations. So this is so the example that I always use is one person gains a mutation that is the best thing to help people survive living in a city, in a smog-filled city. Now that person, they get older and they haven't had any children yet, and this is the best allele. This will help everybody. Whoever gets this will have more children, and they'll keep passing it on. But it's one person has it. That person walks out and gets hit by a bus. That allele is lost. So a random event, usually an environmental effect, something causes the loss of an allele, and that's going to be more pronounced on smaller populations. And so if we look at genetic drift, it can result in one allele becoming fixed if only that one allele exists, or it can be lost. And this is all due simply to chance. So there's this figure from your book that shows a simulation of genetic drift and what happens. So this is a pretty ugly looking graph. We have generations going from on the x-axis, and the frequency of the big allele on the y-axis. And in all these cases, we start out with the frequency of the allele. The big allele is at 50%, so right in the middle. And each of these lines represents a different population, where n is the starting number of individuals in that population. And so we have a whole bunch where there's 20 individuals, all these, and we have one where there's 1,000 individuals. And what you can see for those populations that have 20 individuals, it may take a little while, but in these two cases, the purple and the blue here, eventually the big allele becomes fixed. In the green, red, and orange, eventually the allele is lost. In the green one, it goes up for a little bit and then comes down and is lost. But what you see, so that's what happens with small populations of small size. Different things can happen due to random events. When you look at the population that has a lot of individuals, has 1,000 individuals, well, it'll go up and go down, go up and go down, but it's staying around the midpoint. Now, it looks like it's heading towards fixation, but this would be over a long period of time, and the changes are less dramatic. And as we've seen with this green example, where it goes up and it comes down, well, this could happen for this one too. Maybe when we get further out to the right, this will come back down and get more stabilized around 0.5. But the changes in each generation are very small. And so one of the things that events that can happen that cause this is the bottleneck effect. And this is a population size can be reduced dramatically by a natural disaster. And so a disaster will randomly eliminate individuals regardless of their genotype. So it doesn't matter what the genotype is, everyone has an equal chance of surviving or not surviving. And so this period of this sort of disaster is called a bottleneck. And that's sort of and this bottleneck is when the population size gets very small and becomes susceptible to genetic drift. And so that's sort of shown here. So here we have a very large and genetically diverse population. And that's shown by the different individual balls that are of different colors, blue, red, and green. Something happens, some catastrophic event, where most of the individuals die and only a few survive. And what we can see is that mostly, in this case, there are individuals that have this blue phenotype, one with a red phenotype, and none of those with the green phenotype. So these survive this bottleneck, and so the population is very small. Then the population begins to grow again. And as it grows, well, some of the diversity has been lost. So genetic diversity is reduced. We've lost this genotype, this green genotype for these ones that are shown here. And now we have the population is expanding, but the genetic that existed in the original population does not exist in this new population, derived from this original one, but having gone through this catastrophic bottleneck effect. The other way so to think about this is it's called the founder effect. That if you have a bunch of individuals that exist in, again, I'll use this example of mainland, and a very small group move to an uninhabited island. Well, now all of a sudden, there are very small individuals, so the genetic diversity is most likely going to be less than what is, exists in this much larger population here. And while on this island, if one individual has a particular trait and they die, well, they also, whatever traits they have are now lost. So they are very susceptible to this random genetic drift. Any questions so far? All right, so the last of these is non-random mating. And so in most cases, mating is random in different aspects of different organisms. Mating will be random. An example of non-random mating is inbreeding. That's mating between individuals that are more closely related than any two individuals drawn from a population at random. And so we think of it as mating between relatives. And so this is, I don't know if it's been off for a while, but if it's very Games of Thrones going on here, if you're into that show. Here we have individuals that have an allele for a rare recessive trait. Now, just to remind you, this is not a symbol that I use too much. This represents an individual that is heterozygous and doesn't have the trait. So we're looking at a trait that is rare and recessive. So the phenotype of these individuals are identical, but their genotypes are different. This person is heterozygous and is a carrier. This person is homozygous and doesn't have the trait. Right? Neither of them have the trait. So in this mating, since it's a recessive trait and it's rare, this person is going to be homozygous. Their offspring, none of their offspring will actually ever have the trait. And so here we're just showing two of their offspring who have the trait, and they go and they mate with individuals who again are very, very unlikely to have this trait because it's very rare and it's recessive. So they're not even going to have an allele for this. And so their chance of having an offspring that actually has this trait is zero. What happens if these two first cousins mate? Well, all of a sudden now they are both heterozygous, and so in order we know from heterozygous matings there's a 1/4 or 25% chance of offspring having a recessive trait. So mating between these related individuals now gives a chance of their offspring having the trait, which didn't exist when you were mating with individuals from outside this family. So inbreeding increases the homozygous individuals in the population, and a completely inbred population theoretically will consist only of homozygotes, and it reduces the genetic diversity. Now, this has been a good thing for crop development. So we talk about Mendel's work. You have these true breeding strains. Well, that's a form of inbreeding. The individuals were picked and mated to give particular traits. Mating was non-random, and that's how the different strains, different true breeding strains were allowed to persist for so long. Another thing that we see this with is with sort of animal breeding, getting purebreds. And so these are purebreds are animals that are bred with closer relatives, and so that they are always give the same trait. Now, what's happened is that any traits that are sort of negative start to pop up, that are rare recessive traits, which normally get diluted out in a population. We start to see. So when you start looking at purebred dogs, like the golden retriever, it has a significant risk of cancer, skin allergies. It gets hip pain from joint malformation, and they have heart. They can have potentially fatal heart defects. Bulldogs, they have these weird sort of elbow and joints that may be painful and malformed. Their eyelids sort of droop and can irritate the eye. They have extreme difficulty exercising. So people like to have these, sort of purebred animals. A lot of these animals are not really doing so well. They have an abundance of health problems, and this is really obvious in the Scottish fold cat. This suffers from osteochondrodysplasia. It has shortened limbs. It walks abnormally. It has a very stiff and painful tail, and they develop arthritis at a very early age. And it's so reliable that these cats are actually used as a model to study human arthritis. And so there's been a lot of sort of ethical questions that are being raised about whether or not these, this sort of breeding should actually be allowed to occur. Now, these individuals, if they mate with sort of, outside of this, with different individuals, different breeds, or those that are not purebreds. Well, a lot of these characteristics that are recessive will then be hidden by the alleles coming from individuals that are not in these populations, and their offspring will most likely be healthier. Any questions on this? So there have been good reasons for non-random mating, and then there are some, in my opinion, not really good reasons for non-random mating. Okay, so why is genetic diversity important? And so that's what this non-random mating sort of eliminates that. Well, I've given an example here of why it's good to have some diversity, sort of mask many of these negative attributes that exist. But so you get lots of biodiversity, and this biodiversity helps a population adapt to environmental changes, so conservation, so that the population will persist. Okay, so the example that I use here is, so we have some individuals that, population that we have some heat-resistant individuals, so there's variation in this top population, and in this bottom population there's not as much variation. And then we go through some sort of catastrophic event, in this case, rising temperatures. In this first one, the top population, well, some of those individuals that are resistant to higher temperatures may survive, and those that aren't may die off. But the bottom line is some of the population survives. In the bottom population, if none of these individuals can survive this, they're going to go to extinction. All right. Okay, so I'm going to, we have about half an hour left, and I'm going to talk about sort of another topic to do with population genetics. And now this is really where I started to learn more about this, is where I started to really get interested more so in population genetics. And so we're going to talk about a particular technique. So we've talked about Human Genome Project, and then we started to sequence and determine the genomes from multiple individuals. Talk about HAP maps in a little bit, and sequencing 1,000 genomes. This is now quite some time ago, and so there are more and more genomes being sequenced. But the technique that I'm going to talk about today is this genome-wide genotyping, or GWAS. So this is genome-wide association studies, and the purpose of this is to compare genetic variation with different disease states and traits, and ask, is there a correlation between the variation and a disease? So you need a lot of genomes sequenced from a large number of people so that you can identify things called single nucleotide polymorphisms and compare them with people who have a particular trait or disease with those who don't. So let's talk about this single nucleotide polymorphisms. These are called SNPs for short. So it's a site in the genome where an individual member of a species differs by a single base pair. They occur every, 1 in every 500 to 700 base pairs in the human genome. There's about 10 million SNPs in the human genome. They can be in coding regions where they may alter function, or they can simply be close to a gene, and they will act as a marker for that gene. So if there's some, there might be another difference that is not noticed by this as much, and that trait, these will act as a marker for that trait. And so we get a thing called a haplotype, and it's a set of SNPs and their genetic variants observed on a chromosome. All those words, hopefully these pictures will help. So here we have chromosomes, it'll be chromosome 1, from different individuals A, B, C, and D. And so most of the sequences on a chromosome between individuals is exactly the same. Those are shown as these white letters. But there are places where there are differences. These are the SNPs. Now, haplotype is taking these SNPs and sort of having these different alleles from a chromosome, sort of defining them. So this haplotype A has most of the sequence is identical, except in the 3 positions we're considering here. This is the sequence you get from those 3 positions. B has a different sequence. C has yet another sequence, and as does D. So there are 4 different haplotypes here. So all we've done is extract the useful information from the SNPs that allow the distinction of each chromosome. And so what we'll do is we can put these different SNPs onto a microarray and sort of analyze who has these different variations. Okay, in a more general sense here. We have individuals that have particular traits versus those who don't. It could be a disease or it could be any trait. It's easiest to talk about this in terms of disease. So we look for variants or haplotypes associated with a disease. They're going to be found in higher frequency in those that have it than in the controls. So this is the group that has the disease trait. This is the group that does not. You need a comparison group. So if we look at these different chromosomes here, and we look for a particular SNP, these are these red spots right here, these red bars, and that's the SNP we're looking at. Those that have the particular disease, in this case, we have 12 individuals that we looked at, and 7 out of 12 have those, or that's 58.3% of the individuals have the SNP. If we look at the controls and we count this up, 2 out of 12, or 16.7%, have this SNP. So this variant, is more prevalent in the individuals who have this disease. So in this example, there are more red SNPs in the cases sample than in the control sample. But the point I want to make here also is that both groups can have this particular variant, this SNP, but it's going to be more frequent in those that with the cases, those with the disease. And so it occurs in 58% of individuals here, as opposed to 16%, 17% in the controls. Does everyone follow this? Any questions on this? All right, so just sort of a way to look at this is most of these variants that occur are going to be the same between the disease and the control cases. So most of these, so I've highlighted in magenta and bold, just a demonstration of different positions that can vary. Right, so here we have disease cases versus controls. And if you look, at this position, these will have A or C, about the same numbers in the disease cases control. This position will have G or T, in about the same frequency in the disease cases versus the control. But there is one position in here, and now highlighted in blue, where in the disease cases, there's mostly a G. Not 100%, but mostly a G. And in the control case, there's mostly a T. So there's the associated of these SNPs vary between the disease and the controls. Right? And this is an example that I made to sort of illustrate this point. It's oftentimes not as clear as that. Okay, so what do we do with this? So we'll you get these SNPs get put on a microarray. And you have different, this is obviously a small subset of a microarray, and we look for again variants that occur more at higher frequency in the cases versus the controls. And so this spot here is highlighted relating to this thing over here. All right, so the first step is you isolate the DNA from different individuals, and you put them onto these microarrays and see, well, what SNPs occur. How often do certain SNPs occur in the cases versus the controls? But then you need a statistical way to show that it's actually significant. this difference is significant between the cases and the controls, and that's where this graph comes in here. I'll explain it on the next slide. Anything? Does everyone understand up to this what we're talking about here? Right. So these are one of my favorite kind of data to look at. I think they're called Manhattan plots because they resemble the skyscrapers of the Manhattan skyline. And this is a really a first step at looking at the data that comes out of these a GWAS experiment. And so what do we have here? So on the X chromosome, we have on the X axis, we have all the chromosomes shown here. And in this case, they're all color-coded, and the number of the chromosome is shown underneath them. Remember, these are the bigger chromosomes. They're wider than the ones at the end. Now, these blocks of color are actually thousands of dots because each SNP is represented by a dot. You can see them when they start to stand out. So, obviously here, you can see a bunch here. There's one there. But when they're all sort of, jumbled together, they just look as blocks of color. All right, so that's the x-axis. The y-axis is now the negative log of the probability of the significance that there's a significant difference between the cases and the control, the disease state and the control state. And so the higher that you go up here, because it's a negative log, it indicates that it's the greater chance that this is a particular SNP is associated with a disease, and it's not due to random chance. So this means the higher it is, the more significant. So we're looking at a probability here. And again, just to remind you, each one of these dots is a different SNP. And so what you can see is that we have certain lines going horizontally here. These are cutoff values, and these are used to sort of define, well, what's truly a significant hit versus something that's could be a false positive. And there are oftentimes more than one cutoff. There's usually a very stringent cutoff, which is sometimes like 10 to the 7th or 10 to the 8th. So that's this upper line here. That SNPs that are have a significance above that line, or that appear above that line, are considered highly significant. Then there's oftentimes a sort of a lower cutoff where there are some things down here. These might be important too. They don't have as great as high a statistical significance as these other ones, but they might be important. Right. So what this does is this gives you positions in the chromosome to start looking at. And and you can look at these. You can then take these and look more closely at what's at this region. So here's a bunch of SNPs that are fairly close together. You can look at these different regions and see what genes are there and see if there's anything that makes sense, or you can start designing experiments. And so this has been done for lots of diseases. So a paper came out quite some time ago in 2007. They looked at all these different diseases. They had 14,000 cases and 3,000 controls for these. And they found many different sort of SNPs that had significance values above 5 times 10 to the minus 8. So these are shown here in green. And so these give a nice starting point for all these different diseases. Like, well, maybe we should look at what's going on here on chromosome 9, or we should look at chromosome 5 and chromosome 1, and it looks like chromosome 2 over here for Crohn's disease. For rheumatoid arthritis, there seems to be a couple things. One here that's probably really important on chromosome 6 and something down here. Sorry, this is type 1 diabetes. The name is below it. And you can see that there are some similarities between that and type 2 diabetes, but there are some points that are distinct for between these two forms of diabetes. So there are so this is a good starting point for sort of looking through a lot of information and to determine what genes might be important.
Okay,
So, yes.
Oh, sorry, I had a question.
Yeah.
The y-axis indicates the probability of an SMP being associated with a disease, correct?
Exactly.
Okay, so how is the probability itself calculated?
So there's a lot of factors that go into that. That's so I can't give you the statistics on that. If you've taken biostats, they can probably Dr. Farrell can probably have give you a better description of how that is. So I don't know exactly how they go through. It's going to be obviously what's the sort of percentage difference, and there are other factors that are involved in that.
Okay.
Yeah.
Okay. Thank you.
Yeah. It's complicated beyond what I know.
Okay, but for the sake of this class, we just have to understand the what the axis or the axes.
Yeah. So like if you were to look at this, you say where would you find a gene that might be important for bipolar disorder? You look through this was actually kind of complicated because there's some green dots here. For coronary artery disease, you say, oh, there's something on chromosome 9. You would say, well, how many different sort of regions might be important. And here it looks like there's really one and maybe a couple others. Way of interpreting this, like being able to interpret these graphs to sort of look at what regions might be important.
Okay.
Yeah.
Okay. Thank you.
Yeah, and I don't know why I shouldn't look into this, but chromosome 9 is always missing a part, and so is chromosome 1, and I don't know why that is. Why there are no SNPs in those regions. I noticed this last night. Anyways, that's an aside. Okay, so we're going to talk. I'm going to give a quick case study here that's sort of relevant to today. And so using this kind of analysis for COVID-19. And so this came out, looks like in October of this year, although I saw another reference with the same title that came out in June, so I'm a little confused by that, but this is New England Journal of Medicine. And one thing I want to point here is that there's a severe COVID-19 GWAS group. That is this is the author of this article. And it looks like I thought it's like a block of like 100 different people on this. Okay, so what did they do? Is these people took individuals from Italy and Spain, and it seems like it occurred back in around April. I was having a hard time figuring out exactly the exact dates that they did sampling, and they looked at people hospitalized, patients with COVID-19 that were hospitalized, and they tested positive. So they had a PCR test that showed that they had the virus. And then they compared that to a control, which is a bunch of randomly selected blood donors, and there were a few other groups that were included in there. Right, so they had their cases, which is on top, and their controls. And so when they looked at this, so here is our GWAS plot that we just looked at. They noticed that when they set a pretty reasonable cutoff area, that there were two regions, one on chromosome 3 and one on chromosome 9, that showed that there was a statistical significance of these SNPs being more prevalent in the disease individuals versus the control individuals. All right, so what's going on here is this region, which is chromosome 3. So you may remember sort of the naming of different regions on chromosomes. This would be chromosome 3, and a section on the petite arm of chromosome 3, 21.31. So it tells you where this is located on the chromosome. And in this region, there are 6 genes, some of which have have I left the word out here, have a association with COVID-19. So this top one listed here, SLC 6820, is something that's known to interact with the ACE2 receptor, which is the receptor that COVID 19 that it actually recognizes to gain entry into the cell. And this CXCR6 regulates the location of immune cells, T cells, into airway in response to airway pathogens. So it's taking effect in the lungs, and some of these other genes also have effects in the lungs, as well as some of the genes that are outside of this region, sort of on the boundary. So this seemed, an interesting. It not only that, so they found this region. It was interesting to find that, but it's also interesting to see that there were some genes that seem like they could really have, based on what we know, an effect on whether or not how susceptible a person is to this disease. Now the other one over here, ABO, is the ABO blood type region, and so this is looks like it's on chromosome 9 because it's gray. And this has been implicated in SARS, and so I'm not using these SARS-CoV-1 or 2 designations because this would be SARS-CoV-1 and this is SARS-CoV-2 is COVID-19. So in SARS, which is I forgot when they had that outbreak, but some time ago, and COVID-19, blood type has been sort of implicated as having an effect on infections. And this study suggested that type O blood is associated with lower risk infection. So if you're interested in this, you should definitely go and take a look at the ABO blood types that we've talked about previously. And then I found it, so another article, just now this is not a GWAS article, but this is just sort of looking at the different blood types and what effects they have on infection. And so here they suggest that type O is associated with lower infection risk. And so in this other article, they suggest the same thing, comparing how many. So they have two groups here. These are the individuals that have the SARS-CoV-2, COVID-19, and the reference population. So this was done in Denmark, and it took about 38% of the population of Denmark as their reference population. And so what they found is that fewer COVID-19 patients with type O blood than their control group. And this was found to be statistically significant. The thing that we don't really mention is that there seems to be a statistical significance also that those with type A are slightly more prevalent. The other two, B and AB, did not show any significance. There's no significant difference between the reference population and the population with COVID-19. And so there's evidence that SARS, not COVID-19, but there's been done with SARS in vitro, so biochemical assays, that SARS can be glycosylated by the glucosal transferase from a blood type. So we've talked about this. So not only can the glucosal transferase put its mark on the human cell, on the H substance, it can also put a mark onto the virus itself. And that when you have antibodies that are against the A blood type, they can neutralize the SARS virus. And so they suggest that this could also be true for anti-B antibodies if they did the same thing, which hasn't been done yet. And they suggest that this may be a reason that some individuals may be able to resist infections slightly better. Okay. All right, now to sum up my last slide, is just to sort of think about, sort of this technique and things that can be done with it, and how powerful it really can be. So lots of diseases, you can have a certain disease, so it's considered a certain phenotype. But the underlying reasons for having that disease can be different. Can be different. They can have different. You can have a different genotype that gives the same disease. So if you have individuals, as an example here, a bunch of individuals that have this disease, if you determine the individual SNP pattern with each of these, or the SNP pattern with each of these individuals, you might be able to sort of break them down into different groups based on the profile that they get from this sort of GWAS, the SNP analysis. And so you can have these different groups. Now, these different groups can have the same disease for a different reason and may require a different treatment. And so you might be able to find, a correlation between a particular treatment and a particular group, and you can tailor that treatment now to you can do SNP analysis on individuals that not involved in this study, who have this disease, and determine like, oh, they have this SNP profile. Treatment A will work better than treatment B for them, whereas another individual will have a different SNP profile, and maybe a different treatment will work better for them. So this is a way of developing more personalized treatment. Okay, so that's what I have to say today. And open up to, we have a couple minutes here if anyone has any questions about this. Like I said, this whole sort of the GWAS analysis, when I started really learning more about this myself a few years ago, really sort of started piquing my interest in population genetics and how it can be used, to learn a lot of different things and sort of to help in this sort of aspect, tailoring treatments to different individuals based on their genotype. Okay. All right. All right. So I have a question to just go over the Manhattan plot one more time, and I will do that. If you're satisfied with what we've talked about today, please feel free to go and enjoy the rest of your day. And if you want to stick around and listen to my explanation again, feel free to do so. Okay, I'm going to go back to my original. Yeah. Okay. All right. So just the Manhattan plot. So so I'm going to go through it again, sort of in detail. What we have on the x-axis, every single SNP in the genome is represented as an individual dot across this x-axis as its position in the genome. So this gives the position within the genome. Now, and each SNP is sort of, each chromosome, all its SNPs have been sort of, clumped together, and there are so many in this region here, in this blue region, that you can't see the individual dots. Right, so that's the same case for all of these. And so imagine, this represents an entire chromosome. That's a very small space for all the base pairs that are in that chromosome. Right, so you're going to get everything's going to get very compressed. Right, this is going to be thousands of these SNPs. So that's what's going along the X. So you're breaking down the SNPs by where they're located in the genome, further breaking it down and actually which chromosome they are. So person doing this will know exactly what this dot is. Right? They're going to know what that is. And it's not just randomly put down. It's put down in a very orderly fashion. And you can zoom in on these, and you can get, you can determine what these positions are. Okay, so that's how everything is organized this way. Going on the y-axis is looking at the statistical significance that there's an association of a particular SNP. I like this one because it stands out and it's really high up here, with the disease state. So it's more likely to occur, the SNP is more likely to occur in the individuals who have the disease than in the control group that do not have the disease. So this is a negative log 10 probability. So that means that the higher the number you have, the more likely that there's a significant association, and that's not just due to random changes. So these are all, again, all different positions, and now they're not, they line up really nicely here. They're not all on top of each other in reality. They're across this chromosome. This is chromosome 19. They're across the chromosome, but because this is so squished in here, it's so compressed, They all, there's a lot of things that are very close to each other. So there's a lot, for this sort of random example disease here, there's a lot of places, a lot of SNPs on this chromosome 19, which are significant. And so this cutoff, so when determining something is significant, there are multiple ways to do it. Usually there's what's come out, there's a certain probability that's sort of accepted as convention. That's now these have been done. I don't know how many times these Manhattan plots have been performed, but they're quite common now. And so they know that there's usually somewhere around 10 to the minus 7 or higher. There's most likely it's a significant observation. So that means that there's a high likelihood that this spot has something to do with whatever the random disease that we're talking about here. The trait is going to be associated with the trait, this position in the genome. It doesn't mean that's a particular gene. It means that it's just a difference between it's this polymorphism, a difference that can occur when you see one particular kind of difference. In this case, that particular kind of difference is very likely to be associated with having this disease. So it brings you to a region of the DNA. It doesn't necessarily bring you to a specific gene. So that's, but it gives you an idea of but you can then look at this and say, well, what genes are on the chromosome in this region? And maybe there's only one gene and you have found it. Maybe there's more than one and now you need to look at those. But it's a way of reading through all of the chromosomes, the entire DNA, and finding a few positions that have a high likelihood to be important for whatever you're interested in. And so, so this is a very high stringent cutoff. This means that this is really likely to be important. This lower one here says, like, well, there's the chance that it's important, but not the chance that it's not due to just random a random association is lower, but still pretty high, and way above. You can see this would be level of background down here. You see all these sort of these blocks, and so this will be probably your somebody who is trying to understand what genes are involved in this disease would say, I have 1, 2, 3, 4, 5 regions that are highly significant. I'm going to start with those and investigate what's going on with those. Then they might say there are an additional 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. There's going to be 12 more, somewhere between 10 and 15 more regions that fall between these two lines. That they say, we may want to look at those too. And so that, in essence, is what a Manhattan plot does for you. It's a way of statistically looking at differences in SNPs between individuals with a particular trait you're interested in, whether it be a disease or something else. It doesn't have to be done just for humans. It doesn't have to be done just for disease. It could be eye color. It could be flower color or plant height. It can be done for different organisms, and has been done for different organisms. But it gives a way of looking through all this data and coming and narrowing down to a couple features, a couple places, to really look at. It's really powerful, because that's one of the biggest things is finding what's in the genome, making sense. We talk about genomes, making sense of the genome is difficult. Okay, all right. So we're at 12. Hopefully that answered the question. And for those of you who stuck around, hopefully it helped clear things up. I just like the way these look. I really like them.